we will look at the processes involved in tubular reabsorption and secretion. We will look at some of the major substances which are reabsorbed and secreted along the tubules. We will understand the concept of tubular maximum, renal threshold, looking at glucose as an example. Then we will look at the renal handling of certain substances like glucose, amino acids, sodium, potassium, etc. And towards the end, we will look at the feedback mechanisms that control the glomerular filtration in response to the tubular function. Do a quick recap of what you already know. As you all know, kidney acts as a filter, allowing excretion of waste products. Production of urine is the result of three processes: filtration, reabsorption, and secretion. Filtration is non-specific, producing a large amount of filtrate. While passing through tubules, most of the water and large number of substances are reabsorbed and returned back into the circulation. And certain substances are actively secreted along the tubule, mainly organic acids, and then as a result, urine is produced, which is then transported from the kidney through ureter into the urinary bladder for excretion. Looking at the nephron, which is the structural and functional unit of the kidney, nephron is simply a parallel network of tubules and blood vessels. Now the tubules are shown here. It begins with a cup-like structure, which is called Bowman's capsule, followed by proximal convoluted tubule, followed by the loop of Henle, which is continued as distal convoluted tubule, becoming collecting tubule, and then joining together to form a collecting duct. Now if you superimpose the blood vessels onto the tubules, this is how they appear. So blood flows in through the afferent arteriole into the glomerulus, which is a tuft of capillaries, and this is where filtration takes place. After filtration, the remaining blood flows through the efferent arteriole, and this efferent arteriole follows the tubules, and after reabsorption and secretion, it will become the renal venule, leaving the kidney as the renal vein. Another important point to note here is that usually when nephron is drawn, it is drawn in a schematic way. But if you look at the actual anatomy of nephron, the distal convoluted tubule passes very close to the glomerulus, actually coming in contact with the afferent and the efferent arteriole. The cells in this region are specialized to form what is known as the juxtaglomerular apparatus, and this has a very important role in the regulation of renal function, as we will see towards the end of this lecture. Now after this, I'll just would like to emphasize on the factors that regulate glomerular filtration, as you have already covered those. As you know, the blood is flowing in through the afferent arteriole. In the glomerulus, you have the production of filtrate. The remaining blood flows out through the efferent arteriole. Now imagine if the afferent arteriole is constricted, it will reduce the hydrostatic pressure in the glomerulus, which will reduce the GFR, and also because of afferent arteriolar constriction, there will be reduction in the overall renal blood flow. If there is constriction of efferent arteriole, what will happen? Interestingly, GFR will increase, but renal blood flow will be decreased. So if I put it in a table here, constriction of afferent arteriole reduces both renal plasma flow and GFR. Constriction of efferent arteriole reduces the renal plasma flow but actually causes an increase in GFR. If we look at the filtration fraction, which is calculated by RPF and GFR, constriction of afferent arteriole will have no effect on filtration fraction, but vasoconstriction of efferent arterioles will actually increase the filtration fraction. Now, just to give you a clinical application of this, as you know, angiotensin II and sympathetic nervous system can cause vasoconstriction primarily of the afferent arteriole. So when this happens, it will reduce renal blood flow and also it will reduce the GFR, which could be detrimental for the kidney. So in order to counteract this, kidney has auto-regulatory mechanisms. Prostaglandins play a major role. So they are released locally and they nullify the effects of sympathetic nervous system and angiotensin II, thus maintaining a good GFR. Sometimes non-steroidal anti-inflammatory drugs, if given in high doses, can block the production of these prostaglandins, making this person prone to develop renal failure. Now, after reviewing some basic functional anatomy and some basic factors that regulate GFR, we'll be looking at the tubular transport now. Now, let's imagine 4 possible scenarios. Looking at the figure A, there is a substance which is freely filtered at the glomerulus. There is no reabsorption and no secretion of this substance. Alright, so this substance is freely filtered but not reabsorbed and not even secreted. Substance B is a substance which is freely filtered, but part of the filtered load is reabsorbed back into the blood. So what we can see here, substance A is totally dependent upon filtration, but substance B is dependent upon filtration plus reabsorption. So the final amount that appears in the urine will be much less than the amount that was filtered. Moving on, let's think of another substance, substance C, which is freely filtered and then it is completely reabsorbed, which means nothing appears in the urine. All right. Moving on, look at the 4th possible scenario. There's a substance which is filtered. There is some amount which is still remaining in the blood in the efferent arteriole, and when these blood vessels pass along the tubule, all of it is secreted into the tubule. So this substance is completely cleared by the kidney. So the renal vein, the blood in the renal vein which is leaving the kidney should be completely free of this substance because kidney has completely cleared it off. All right. Now let me put it in a different way. So this is a diagram showing you the percent filtered load of a particular substance remaining in the tubules. So there is a substance which is say the green color. Okay, it is filtered 100% and it is excreted in the same amount in the urine. So this is the example that I showed you, the substance A, filtered, neither reabsorbed nor secreted. So this substance can be used to calculate GFR, and as you all know, a perfect example of this one is creatinine, which is an intrinsic substance, or inulin, which can be administered intravenously and can be used to measure GFR. Now look at the substance which is shown in the blue color curve. If you look at it, it is filtered and then it is being excreted or secreted, and at the end the amount that you have in urine is much more than the amount which was filtered. So this substance is filtered and then actively secreted. So in the urine you have much higher concentration than the concentration that was filtered at the Bowman's capsule. Looking at the third one, which is shown in red color, this substance is filtered and then quickly reabsorbed in the proximal tubule, so nothing appears in the urine. Now example of this substance would be glucose, and also to some extent those amino acids which are filtered will be quickly reabsorbed. Alright. Now looking at the tubular reabsorption, as you all know, filtration is a non-specific phenomenon. Whether a substance appears in the urine or not will depend upon the tubular modification, which is reabsorption or secretion. Now tubular reabsorption is highly selective. The kidneys regulate the excretion of solutes independently of one another. This is a capability which is essential for precise control of the composition of body fluids. Now what we will do, we will look at a schematic diagram of renal tubule starting from Bowman's capsule. The first one is proximal convoluted tubule, followed by the loop of Henle. The loop of Henle has three parts. The descending limb of loop of Henle is a thin tube. It loops around and becomes the thin ascending limb, but the later part of the ascending limb becomes a thicker tubule, which is called thick ascending limb, which will then continue as distal convoluted tubule, which will then become the collecting tubule and collecting duct. Now, putting this into the structure of kidney, the proximal tubule and distal tubule are present in the cortical region of the kidney, while the loop of Henle goes down into the renal medulla and then loops back. The collecting duct is also present. It starts in the cortex, goes to the outer medulla, all the way into the inner medulla, and then join together to form the renal pelvis. We will now zoom in into different parts of the tubule and look at the processes which are occurring there. Now, normally 65% of the filtered load of sodium and water and a slightly higher percentage of slightly lower percentage of filtered chloride are reabsorbed by the proximal tubule. Almost all of the glucose and amino acids are reabsorbed here. The proximal tubule is also an important site for secretion of organic acids and bases such as bile salts, oxalate, urate, and catecholamines, and also most drugs are secreted here. Moving on from proximal tubule to the loop of Henle. Now this is a bit complicated, but let me take you through step by step. The thin descending segment of loop of Henle is highly permeable to water and moderately permeable to most solutes. So what happens here? It mainly allows simple diffusion of substances through its walls, and about 20% of the filtered water is reabsorbed here. Now looking at the thin and thick ascending segment of the loop of Henle, this segment is virtually impermeable to water. Now this is a very important characteristic that is, as we will see later, is very important for concentrating the urine. Now the thick segment has thick epithelial cells that have very high metabolic activity and are capable of active reabsorption of sodium, chloride and potassium. Now around 25% of the filtered loads of sodium, chloride and potassium are reabsorbed over this part of the tubules. Now moving on to the distal tubule. The distal tubule, you have to appreciate a fact that the very first part of the distal tubule comes into contact with the afferent arteriole, as I showed you in the picture at the beginning of the lecture. So it forms the juxtaglomerular apparatus. Moving on from there, the first half of the distal convoluted tubule is much similar to the ascending limb of loop of Henle, and the second half of the distal tubule is much similar to the collecting duct. Now basically this part of the tubules, which is the distal convoluted tubule, is composed of two types of cells. The principal cells, which reabsorb sodium and secrete potassium. There are also intercalated alpha cells, which reabsorb bicarbonate and secrete H+. So these cells are important in acid-base regulation. Principal cells are under the effect of an hormone which is called aldosterone. As you all know, aldosterone causes sodium reabsorption and potassium excretion. And this is what is shown over here. Now moving on to the collecting duct. It also has the principal cells and intercalated cells as I described for the distal tubule in the previous slide. So what happens basically here, that there is 5% of sodium chloride will be reabsorbed here, and small amount of water is also reabsorbed, and this is under the action of a hormone known as ADH, which we will study later. So the permeability of the medullary collecting duct to water is dependent upon ADH, which we will study in the second lecture, how kidneys are able to conserve water and produce a concentrated urine. Now a general summary of what we have covered in the past, in the previous few slides. Filtration occurs at the Bowman's capsule. In the proximal tubule, there is a lot of reabsorption and some secretion that takes place. Almost 65% of reabsorption of water and solutes will occur during the time the fluid is passing through the proximal tubule. The loop of Henle, the descending limb is very similar to the proximal tubule. Some water reabsorption and solute reabsorption occurs here. The ascending part of loop of Henle is impermeable to water, but it actively reabsorbs solutes, mainly sodium. Moving on to the distal tubule, it is much similar to the proximal tubule, but the magnitude of reabsorption and secretion is much less. Collecting duct is also involved in some reabsorption and secretion. Although the magnitude of reabsorption and secretion is much less in the distal tubule and collecting duct, but it is physiologically very important because it is variable. and it responds to different hormones, which means it responds to the needs of the body. Now, moving on from here, let's add another layer to what we have covered until now. This will help you understand the renal pharmacology. Now, proximal tubule participates in the majority of solute reabsorption via isoosmotic reabsorption of solutes and water, which means sodium is actively reabsorbed and water follows the sodium. Now, how is sodium reabsorbed? There are different transporters and different mechanisms involved here. Now, this occurs through the action of sodium and glucose co-transporter. Sodium actively gets reabsorbed and glucose follows it. Same thing happens for amino acids. Sodium actively reabsorbed and takes amino acid with them using special carrier proteins. Also, there is reabsorption of phosphate together with sodium, plus there is some excretion or rather secretion of H+ or organic acids also linked with sodium reabsorption. Now, what are the factors that can act on the proximal tubule? Let me give you some examples which are clinically important. Angiotensin II regulates the sodium-hydrogen exchanger, causing increased sodium reabsorption and H+ excretion. The atrial natriuretic peptide, or ANP, generally decreases sodium reabsorption through the proximal tubule. Parathyroid hormone, or PTH, inhibits the action of sodium phosphate reabsorption, acting on the proximal tubule. Now, when we move on to the loop of Henle, it is mainly concerned with concentrating the urine, and this topic will be covered in the second lecture. So we just leave it for now. Moving on to the, just looking at the transporters here, okay. The thick ascending loop of Henle, if you remember, I told you earlier, it is impermeable to water, but it reabsorbs a large amount of solutes. And the main transporter here is the sodium potassium 2-chloride triporter. All right. Now moving on from here, let's see what is happening in the distal tubule. Distal tubule, there is sodium reabsorption together with chloride. There is calcium reabsorption that occurs here, and this calcium reabsorption is under the action of parathyroid hormone, right? And remember, the initial part of the distal tubule forms the part of juxtaglomerular apparatus. Now moving on to the second half of the distal tubule and the initial part of the collecting duct, there are two types of cells. If you remember, there are principal cells and intercalated cells. Principal cells reabsorb sodium through the epithelial sodium channels, or ENaC. This reabsorption of sodium is followed by secretion of potassium through the principal cells. They are also involved in water reabsorption in response to the hormone ADH, which causes the insertion of water channels, and water will be reabsorbed. So principal cells are the site of action of two hormones, aldosterone acting on the sodium and potassium countertransport, and ADH leading to the formation of water channels, which allows water reabsorption from the principal cells. Now, moving on to the intercalated cells, there are two types of cells. I already mentioned the alpha intercalated cells in the previous slides. So these cells are basically involved in secretion of H+. The beta cells are involved in the secretion of bicarbonate. Now these cells are very important in acid-base regulation, and you will be studying them in detail when you do your acid-base regulation. Now let's look at different diuretics which act on these renal tubules, causing diuresis. Loop diuretics act on the sodium potassium 2-chloride triporter in the ascending limb of loop of Henle, blocking the reabsorption of these solutes. When you have more solutes in the tubular fluid, more water will follow the solutes, so there will be increased production of urine or diuresis. Thiazide diuretics act on the sodium chloride channel in the distal convoluted tubule, again leading to increased production of urine or diuresis. Now this whole story looks a bit complicated, and renal physiology is a bit tricky, but I think if you keep looking at this picture and study this in detail, you should be able to master these concepts. As you know, as you can see from these previous slides and the description of various renal tubular segments, the regulation of tubular reabsorption has to be a complex process, and it requires multiple neuronal, hormonal, and local control mechanisms. An important feature of tubular reabsorption is that reabsorption of some solutes can be regulated independently of others, especially through hormonal control mechanisms. Another point to remember here is that reabsorption and secretion are accompanied via various transport mechanisms. There is simple diffusion, there is facilitated diffusion, there is osmosis, there is carrier-mediated transport, there is active transport requiring ATP. So all these different mechanisms are involved. Now we're going to look at the carrier-mediated transport a bit more in detail, because for this type of transport, carrier proteins are required, and these carrier proteins have a tubular or a transport maximum which determines the renal threshold. So in the next few slides, I will try to explain the concept of renal threshold and the transport maximum. For this tubular transport maximum and renal threshold, we will take the example of glucose. Now, why this tubular maximum occurs? As I told you earlier, so that those substances that are actively reabsorbed or secreted, there is a limit to the rate at which the solute can be transported. This limit is due to saturation of the specific transport systems or proteins involved. When the amount of solute delivered to the tubules exceeds the capacity of these carrier proteins and the enzymes involved in this transport process. Let me try to explain this in the diagram in the next slide. Now imagine this is a single tubular cell in the proximal tubule. One side is the tubular lumen, other side is the capillary where reabsorption will occur. So basically what happens that sodium is actively reabsorbed from the basolateral surface of these cells and goes into the capillaries. This creates a deficiency of sodium inside the cell, which creates an electrochemical gradient. So sodium from the tubular lumen will move into the cell. When this sodium moves into the cell, it uses a specific protein which also allows one glucose molecule to move into the cell. So glucose basically follows the sodium and enters the cytoplasm of the cell, and then from the basolateral surface of the cell, the glucose goes out using another carrier through facilitated diffusion. So what is evident here? That sodium is reabsorbed actively, creating a sodium deficiency in the cytoplasm of these cells. When sodium from the tubular lumen moves into the cytoplasm of these cells, it carries a glucose along. And this glucose transport is sodium dependent but insulin independent. So it will not change, the amount of glucose reabsorbed will not change as a response of changing concentrations of insulin in the blood. Now let's look at the concept of tubular maximum and the renal threshold. Let's look at the renal titration curve for glucose. On x-axis, the plasma concentration of glucose is displayed, and y-axis is the filtered load and whether the glucose will be reabsorbed or it will appear in the urine. Now, as you can imagine, as the plasma concentration of glucose increases, the filtered load of glucose will also increase. More glucose in the plasma, more will be filtered into the Bowman's capsule. So basically, and you know that glucose is completely reabsorbed. But imagine that there is a limit which is around 180 milligrams per deciliter of plasma glucose concentration. At this concentration, 100% of glucose will be reabsorbed, but as concentration goes beyond this level, kidney will not be able to reabsorb 100% of the glucose that is filtered. All right. So now there are two terms here. Just let me clarify these two terms because there is some confusion in these. Now, renal threshold refers to the plasma concentration of glucose, and the tubular maximum or the transport maximum for glucose refers to the amount that is being filtered into the Bowman's capsule per minute. So as you can see here, at the plasma concentration of 180 milligrams per deciliter, there is about 350 milligrams of glucose being filtered per minute. Until this point, 100% of glucose will be reabsorbed. But as plasma concentration increases, the glucose reabsorption will not be 100%. So the amount of glucose that is extra, which cannot be reabsorbed, will start appearing in the urine. So let me just repeat what I said. Tubular maximum is the maximum amount of the glucose which can be reabsorbed per minute. So until about 350 or 375, the renal tubules will be able to reabsorb all of glucose. And this usually happens when the plasma level is about 180 milligram per deciliter or 10 millimoles per liter. When this renal threshold is exceeded, glucose will start appearing in the urine. Now let's look at some conditions where this could happen. So as I just told you in the previous slide, the tubular maximum for glucose is between 350 to 375. Mainly in women, it's about slightly lower than men. It is reduced in about 40% of pregnancies. So even with normal plasma glucose levels, glucose may start appearing in the urine. In diabetic patients, also glucose will appear in the urine, but mechanism is different. Very important to understand here. The glycosuria that appears in pregnancies has nothing to do with the renal threshold. It is the tubular maximum which gets lowered, and even below 180 milligram per deciliter of plasma glucose, glucose will start appearing in the urine. So this is a lowering of the renal threshold. In case of diabetic patients, there is no lowering of renal threshold. Actually, it is the amount of glucose in plasma which is much higher than the renal threshold. So glucose starts appearing in the urine. All right. Now let's look at the renal handling of sodium. We have actually covered it previously, but let us put it in one slide together, all different renal tubules, the different parts of renal tubules. Now for sodium reabsorption, this is a global phenomenon. Whether you're talking about proximal tubule, renal, the loop of Henle, distal tubule, whichever part of tubules, this is a global phenomenon. The sodium-potassium ATPase pump working at the basolateral surface actively reabsorbs sodium into the blood, causing an electrochemical gradient within the cytoplasm so that sodium can move from tubular lumen into the cytoplasm. Now, different parts of the tubules use different mechanisms for sodium reabsorption. I'm only mentioning the major ones here. In the proximal convoluted tubule, 60% of sodium is reabsorbed, and it is mainly through the sodium proton exchange. In the thick ascending limb of loop of Henle, 30% of filtered load of sodium is reabsorbed, and it is through the sodium 2-chloride potassium cotransporter. Moving on to the distal tubule, 7% of filtered load of sodium is reabsorbed here using the sodium 2-chloride cotransport channels. In the collecting duct and also part of the distal convoluted tubule, remaining 3% of sodium is reabsorbed, and this is through the epithelial sodium channels. All right. Now there are different hormones that influence sodium reabsorption. There are some which increase the sodium reabsorption, and you all know the hormones. It is renin, angiotensin II, and aldosterone. There is one hormone, however, which causes a decrease in sodium reabsorption, and that is the atrial natriuretic peptide, or ANP. Now those hormones which increase sodium reabsorption will lead to a decrease in the production of urine, and those hormones which decrease reabsorption, like ANP, will lead to an increased production of urine. Factors that influence renal handling of sodium: GFR, which actually determines the amount of sodium that is filtered, is affected by various factors, and you have covered these factors before. You know, glucocorticoids are required to maintain normal GFR. There are two mechanisms which are dependent upon the tubules. So tubules actually talk to the Bowman's capsule and the glomerulus, and tubules can actually influence the GFR, and I will try to explain this in the next couple of slides. In addition to this, the sympathetic nerves also increase sodium reabsorption. They can do it through their direct effect on the tubular cells, or they can indirectly act through releasing renin and activating the renin, angiotensin, aldosterone system. Changes in tubular reabsorption of sodium are more or less constant in the initial part of tubules. It is the later part of tubules which is very sensitive to these regulatory mechanisms, mainly the hormones, and that will determine the final constituents of urine, the osmolality of urine, the specific gravity, etc. Let's look at some other solutes in addition to sodium. Chloride actually follows sodium. They have a very good friendship. They always go together. Some chloride is reabsorbed when water is reabsorbed through the tubules via osmosis. Urea, which is a waste product, is also passively reabsorbed from the tubules. Small amount, as we will see later. Why this waste product is reabsorbed, this has an importance in the kidney's ability to form a concentrated urine, and we will cover this in the second lecture. But mainly, most of urea will be excreted in the urine. Let's look at the renal handling of potassium. More than 90% of potassium that is filtered will be reabsorbed, mainly reabsorbed in the proximal convoluted tubule. Small amounts are secreted in the distal tubules under the action of aldosterone. If you remember, aldosterone causes sodium reabsorption and potassium excretion. The excretion of potassium is never zero, because some amount of potassium is always secreted in the distal tubules. Also, the secretion of potassium competes with H+, thus it has a very important role in acid-base balance, which you will cover when you will study acid-base disorders. Now, moving on to the control, the auto-regulatory control mechanisms within the kidney that can regulate GFR. There are two major mechanisms that I will explain to you now. The first one, which is called Glomerulo-tubular Balance. So imagine a scenario where there is increased filtration of sodium, which means there is increased GFR. All right. When there is increased load of GFR, okay, it is usually due to increased blood flow to the kidney. When there is increased blood flow to the kidney, it means the blood which is flowing in the peritubular capillaries after the efferent arteriole, this blood will have more proteins as well, because the flow has increased, so more proteins are flowing. All right, so there is increased oncotic pressure in these capillaries, which will have increased pulling pressure, so more water and sodium will be reabsorbed. It simply means when there is increased GFR due to increased blood flow through the glomerulus, there will be increased blood flow in the peritubular capillaries as well. There will be more proteins there, so they will have a stronger pulling force on water and sodium, and you will have increased sodium reabsorption. So this prevents sodium loss in urine in cases where GFR is slightly more than normal. The second mechanism, which is called the tubuloglomerular feedback. Now, imagine if there is again increased filtration of sodium, okay, and if it is not reabsorbed very well in the proximal tubules, you will have more sodium reaching the distal convoluted tubule. When more sodium reaches the distal tubule, it causes release of certain vasoconstrictors. If you remember, distal tubule is very near to the glomerulus, forming the juxtaglomerular apparatus. Now, this distal tubule releases a local vasoconstrictor which will constrict the afferent arteriole. When afferent arteriole gets constricted, GFR will drop and the filtration of sodium will also drop, come back to normal. Okay, so again, using this mechanism, the kidneys can conserve sodium, they can reduce the GFR. So now just looking at what we have covered in this lecture today. The filtrate is produced at the Bowman's capsule, which is a non-specific filtrate. It passes through different parts of renal tubules. Different amounts of water and solute are reabsorbed. Certain areas there is active reabsorption, others there is passive reabsorption. And at the end, when the tubular fluid passes through all these various segments of renal tubule, usually 99% of the filtrate gets reabsorbed and only 1% of this filtrate appears as urine that is excreted from the body. So this is all for this lecture, and I hope to see you for the second lecture, where I will try to explain to you the ability of kidney to form a concentrated urine and a few other important concepts. Thank you very much.